Wahoo are fast-moving pelagic fish that grow quickly and move large distances across the Atlantic Ocean. They form an important component of recreational and commercial fisheries and contribute to food security at many Atlantic Ocean islands. However, because they range such large areas of the open ocean, it can be difficult to know whether fish caught in different parts of the Atlantic belong to the same population, or whether there are separate regional groups. This is, of course, an important management concern, as understanding stock structure can and will help improve fisheries management of this important resource. This project will use Otolith Microchemistry to explore issues in connectivity and movement of Wahoo across the Atlantic Ocean. Otoliths are small ear bones found inside the head of bony fish. They help the fish with balance and hearing, but they also grow in layers throughout the fish's life. As they grow, they can take up tiny chemical traces from the surrounding environment. In this way, an otolith can act a little like a natural archive, recording information from the fish's early life through to the point it was caught. Using a core-to-edge approach, a high-powered laser burns the otolith, vaporizing microscopic amounts from the hard sample, where the gases are then analysed by sensitive equipment to measure its exact elemental and isotopic composition. Using a process called laser ablation, inductively coupled plasma mass spectrometry. However, the first step in the process concerns actually getting the fish to sample. These can be from fisheries discards, as all we will be collecting is the carcass of the animal, or from recreational fishers or other means. Once we have the carcass, there are a few things that we need to collect. A sampling and protocol guide has been created and distributed to all collaborators, which includes a printable datasheet to use in the field. An Excel datasheet has also been distributed, where you can copy over your data. The first step is putting down the sample ID, which will connect metadata with samples collected. Then, we collect basic information, such as the capture date of when the fish was caught, the approximate location of capture, lats and longs can be determined later, and the fishing gear or method of capture. Often this will be trolling with lures for recreational fishing, but maybe through other means, such as spearfishing or longlining. This metadata is important because it links each otolith back to the fish and the place where it was caught. Following this, we need to collect data from the fish itself. The first measurement will be the fork length in centimetres. This measurement is the straight line distance from the tip of the snout to the centre of the fork in its tail. This is most accurately measured by placing the tape measure flat and beneath the fish. If the whole animal or carcass is not available and only the head has been saved, you can measure the head length by measuring from the tip of the snout to the most posterior point of the gill cover. Please clearly note HL if you are taking this, or another measurement other than fork length, and note this in the notes column. You can also measure the weight of the fish if it is whole, or the filleted carcass weight. The type of measurement, whole fish, gutted, filleted and gutted, etc., can be noted in the next column. Following this, the sex of the fish and the stage can be noted, if you are confident to do so. We recommend taking a photo of the fish for later reference so that sex and stage can be determined. In this case, please note the specimen ID somewhere in the photo, such as on a piece of paper. Following this, we can move on to the otolith extraction. For this, we will need a saw. A fine-toothed hacksaw is the best option to prevent bone splintering. Fine point tweezers for removing the otoliths. A clean, sharp knife for cutting tissue samples. Vials, such as 2 ml Eppendorf tubes. You will need 3 vials for each specimen, 1 for the otoliths and 2 for clean tissue samples. A Sharpie to put the sample ID clearly on the sample, or a label to put on or in the vial with the sample ID to connect samples with metadata. A step-by-step extraction process starts by placing the fish head on a stable, flat surface. Wipe away any blood or slime from the head with a cloth or tissue paper. The brain is located just above and behind the eyes, so a cut with the saw should begin about 2 to 5 centimeters before the eyes, cutting horizontally if the fish is lying flat. Cut back towards the gill plates, where a downward cut can then be made to remove the top of the fish's head. Don't cut too deeply. You only want to saw through the thick skull plate without destroying what is underneath. A shallow cut is easier to fix than a deeper cut. Removing the head first makes cutting through the skull much easier. The head can be placed facing up, and you can grip the eyes while cutting straight down. To locate the otoliths, you can scoop out the brain with your finger or the back of tweezers. Once all the brain tissue is removed and you are looking at the head straight on, you should see multiple indentations. The central canal sits right in the middle, and this hollow space runs down the length of the spine. The otoliths are housed in 2 small hollow pockets off to either side and just below the central canal. With the tweezers, the semicircular canals can be removed. Then, using the tweezers, the otolith capsule can gently be removed. This clear tissue can be removed by gently holding between your fingers and peeling it away with the tweezers. Place the collected otolith into a vial before moving on to the next one. The otolith can be put straight into a dry vial where it can be stored as is, or a drop of ethanol can be placed into the vial to dry out any remaining tissue. Please note that the vial should be left open for the ethanol to evaporate, or some tissue paper should be inserted to remove any remaining ethanol. Please don't store the samples with any liquids in the vial, as it can degrade the otolith. We recommend collecting 2 clean muscle tissue samples of approximately half a cubic centimetre each. These samples should come from clean, fresh tissue. The cleanest area to collect from an already filleted fish is behind the head, where a small amount of tissue can be cut away, exposing clean muscle, which has not come into contact with dirty knives or hands. The 2 small samples can then be cut out with a clean knife and placed in the vials. One sample, for stable isotope analysis, will be placed into a clean vial and stored as is in the freezer. The other sample, for genetic analysis, will receive approximately 2 ml of 96% ethanol and be stored in the freezer as well. As with the otolith samples, each vial should be marked with the specimen ID. That's all we need from each fish. We are aiming to collect samples from 50 Wahoo at each location, which includes Ascension, St Helena, Bermuda and Turks and Caicos. And we appreciate your support for this project. By comparing otoliths from Wahoo collected in different territories, we can look for regional chemical signatures. This will help us understand whether Wahoo caught around different islands are likely to be connected, and whether current management areas reflect real biological patterns. Each sample is therefore more than just a fish record. It is a small piece of evidence that can help improve our understanding of Wahoo movement, stock structure, and the information needed to manage these species more effectively across the Atlantic.